Did you know that the heredity of microbes is different from ours in another important way? They can inherit genes along many different routes. They can gain genes as we do as copies of the genes of their direct ancestors. This process is known as vertical inheritance. But they can also inherit genes from unrelated microbes through horizontal inheritance. Horizontal inheritance helped scientists discover what genes were made of. In the 1920s, research discovered that if they killed deadly strains of bacteria and mixed them with harmless ones, the harmless strains turned deadly. What's more, when the transferred bacteria divided, their descendants inherited their deadliness. Later, a microbiologist named Oswald Avery and his colleagues isolated the different kinds of molecules inside bacteria to figure out which was the mysterious transforming principle. Through many rounds of experiments, they came down in favor of DNA. It turned out that the bacteria Avery studied were being transformed by taking up loose DNA and incorporating some of it into their own chromosome. They gained genes that they could use to make their host sick. But later research has revealed that horizontal inheritance can also take place by other means. Along with their main chromosomes, for example, microbes often carry ringlets of DNA called plasmids with genes of their own. Microbes will sometimes build tubes that they stick into other microbes, pumping in their plasmids. The plasmid may then float in its new host, or it may paste itself into the chromosome. As scientists examine microbes more closely, they have discovered still more strange forms of heredity. One particularly weird kind of microbial inheritance came to light in the early 2000s as scientists were investigating how bacteria fight against viruses. It turns out that when many species of microbes are exposed to a new virus, they can learn how to stage a swift, precise attack against it. Vertebrate animals like ourselves have the same capacity. When we get attacked by influenza or a cold virus, our immune system can build antibodies that will wipe these strains out as soon as they try to attack us again. Bacteria can't use an immune system made up of billions of cells. Each microbe is one of the cells that has to fend for itself. But they manage this feat all on their own using a system of molecules called CRISPR-Cas. When viruses infect bacteria, they typically land on their victim and inject a string of DNA inside. Many microbes can chop off the tip of this incoming DNA and insert it into a stretch of its own DNA called a CRISPR region. CRISPR is a short for clustered regularly interspaced short palindromic repeats. If microbes manage to survive this initial attack from a virus, they will be equipped to resist the next one. They prepare for a subsequent infection by building a short RNA molecules that match the bit of a viral DNA they grabbed from the first attack. A protein called a Cas enzyme folds itself around the RNA molecule and the two together float off through the cell. If the same strain of virus tries to inject its DNA into the microbe again, the CRISPR-Cas system latched onto the incoming genes. The Cas enzyme pulls the viral DNA strands apart and chops them into the pieces. Shredded into harmless debris, the virus cannot take over the microbe. As a microbe battles virus after virus, it may store away samples from a dozen of its enemies, and when it divides, it passes down this accumulated knowledge to its descendants. When the microbe copies its chromosome, it copies its CRISPR region along with the rest of its DNA.